This educational module equips clinical researchers and trial designers with the knowledge to select and justify pragmatic trial designs that align with their research questions. In this module, we'll start by asking, what exactly are we trying to prove in our trial? Down through the design options that serve that question. We'll contrast the classic individually randomized parallel trial with factorial, crossover, stratified, and cluster design approaches that can be suitable under certain scenarios. In the cluster trial settings, we discuss the importance of identifying the unit of randomization, the unit of intervention, and the unit of analysis, and why they might differ based on the design choices and intervention being tested. Finally, we'll tour a variant of the cluster trial design, the Stepped-Wedge trial design. In any trial, you should start by stating the research question we are trying to answer and the intent we have for the result. The intent might be to prove one treatment is better, to show a new approach is no worse than standard care, or to demonstrate that two strategies are effectively equivalent. This intent isn't just a statistical technicality. It should drive every design decision we make. From how we randomize patients or clinics to the design structure we use and how we plan our analysis, all these elements must align with the trial's intent. Let's clarify these core trial intents a bit more. A superiority trial aims to show that one intervention, say a new drug or care strategy, is superior to another or to usual care. Essentially, we're looking for a clear win, better outcomes with the new approach. In contrast, a non-inferiority trial aims to show the new treatment is not unacceptably worse than the comparator. We do that because the new treatment might be safer, cheaper, or easier to use or implement, so we'll accept it being just as good or a little bit worse within a small margin of difference. Finally, an equivalence trial seeks to show two treatments have no important difference at all, basically that they're interchangeable in outcomes. This often involves demonstrating that the effects of the new and usual treatments lie within a narrow range of each other. With our intent in mind, we next consider the overall trial design structure. The most common design structure is the parallel design, where participants are randomly assigned to one of the study arms and remain in that arm for the duration of the trial. For example, half of the participants receive the new care strategy, and the other half continue with usual care. Pragmatic trials often use parallel designs because they are easier to implement in real-world settings compared to other design structures. We simply need to ensure proper randomization so that the groups are comparable at baseline, and that we enroll enough participants in each group to have the power to detect any treatment effect that may exist. Factorial designs let us test multiple interventions in a single trial. The common example is a 2x2 factorial. Imagine we want to test a new care protocol and a new patient education program at the same time. We can randomize participants to one of four combinations, both interventions A and B. Only treatment A. Only treatment B. Or neither. This way, we can evaluate the effect of each intervention independently, comparing outcomes in groups with treatment A versus groups without treatment A, and similarly for treatment B, all within one trial. This design is powerful if we suspect the two interventions don't strongly interfere with each other's effects. In the absence of interactions between the two treatments, the sample size is the same as that of a standard two-arm trial, powered for a single comparison. However, a larger sample size might be required when you want adequate power to test for interaction effects, or one treatment's target effect is smaller than the others, or the trialist intends to conduct multiplicity adjustments. Another structure you might encounter is the crossover design. Here, each randomized unit gets both interventions sequentially. For instance, in a two-period crossover design, one group of participants starts on treatment A and then later switches to treatment B, while the other group starts on treatment B and then switches to A. Because every participant tries both, each person can serve as their own control. This can greatly reduce outcome variability. Differences are measured within the same individual, which can allow a smaller sample size to detect effects. However, crossovers have important limitations. The condition under study typically requires stability or chronicity, as participants undergo multiple treatments. You also have to worry about carryover effects. What if the first treatment's effects haven't worn off before the second? For that reason, these types of trials often include a washout period between treatments to mitigate this effect. Stratified designs introduce an added layer of control before randomization. Participants are first divided into subgroups called strata based on characteristics likely to influence the outcome, such as age, sex, or disease severity. In some settings, like multi-center individual-level trials, it's also common to stratify by center to account for site-level variation. Within each stratum, participants are then randomized separately. This helps ensure key traits are balanced across trial arms, reducing the risk that random variation introduces bias. However, creating too many strata, especially with a small number of participants in each stratum, can actually worsen the balance and complicate the randomization process. This is called overstratifying, and it's a common pitfall. Two key considerations to keep in mind. First, only stratify on variables that are strongly associated with the outcome. And second, don't forget to account for those stratification variables in your analysis. Otherwise, you lose the benefit they provide. So far, we've assumed individuals are randomized. But sometimes it makes sense to randomize entire groups of individuals or clusters, like clinics or hospitals. There can be many compelling reasons to use a cluster trial design. Sometimes it's the only way because our intervention is something like a clinic-wide training, a new electronic health record alert, or a policy change. You have to implement it by site. In these cases, cluster design aligns with how the intervention works in the real world. Cluster trials can also simplify operations. Imagine trying to run multiple care protocols in a clinic. It can quickly get confusing for staff and participants. It's often much easier if half of the participating clinics do things the new way and the other half continue the usual way. This one-site, one-method approach reduces logistical errors and burden on providers. Also, and related to that, it avoids the potential for contamination, meaning there is a lower risk of participants or providers at the same site accidentally mixing up interventions or influencing each other with knowledge in different arms of the trial. When designing a cluster trial, it's important to recognize that the unit of randomization, the unit of intervention, and the unit of analysis are not always the same, and that's okay. What matters is understanding how they relate to each other and adjusting your design and analysis accordingly. Take this hand hygiene program example. The randomization happens at the ward level. Whole hospital wards are assigned to either the intervention or control group. The intervention itself is delivered to the staff within those wards. But the outcomes, like infection rates, are measured at the patient level. This is a common and valid setup. However, it only works if the trial design includes appropriate administrative and ethical approvals at each level, such as institutional, ward, staff, and patient. These approvals are needed for randomization, involvement, and data collection. And then, the analysis must account for clustering. Cluster trial designs also come with important trade-offs. One of the biggest is the statistical cost. Because participants within the same cluster tend to have similar outcomes, a concept known as intracluster correlation, each person contributes less independent information. As a result, we often require a larger total sample size to achieve adequate statistical power compared to scenarios where an individual-level randomized trial is feasible. Another challenge is recruitment and consent. If individuals are asked to join a study after their clinic has already been assigned to a group, you might see skewed participation. Fewer patients might agree to join if they're in the less desirable arm. And finally, the analysis and interpretation of results from cluster trials is more complex. It requires multi-level modeling and a solid understanding of what kind of treatment effect you're actually estimating. For example, your analysis can provide two different kinds of answers, depending on the statistical method you select. A population average effect describes how well the treatment works on average across all participants and all sites. To estimate this, we often use methods called marginal models, like generalized estimating equations. These methods pool everyone's results together, giving you the bird's-eye view across the entire population. A cluster-specific effect describes how well the treatment performs within a typical site or group. To estimate this, we often use mixed effects models. These models recognize that each site might respond slightly differently, so the results reflect the treatment effect you'd expect within a typical cluster. Next, it's important to consider the level at which you're analyzing your outcome data. When the analysis uses individual-level outcomes, while properly accounting for clustering, you are measuring the treatment effect at the individual level. Using marginal methods here gives you the average effect for all individuals, while mixed effects methods give you the effect you'd see for a typical individual within a site. On the other hand, when you average the results for each site first and then analyze those averages, you're measuring the treatment effect at the cluster level. Now let's look at a variant of cluster trials called the Stepped-Wedge design. In this design, all clusters begin in the control condition. Then at set intervals, say every few months, a group of clusters is randomized to cross over to the intervention. This staggered rollout continues until, by the end of the study, all clusters have received the intervention at least for some pre-specified time. The Stepped-Wedge design is useful when practical or ethical reasons prevent delivering the intervention to everyone at once. It can increase buy-in from partner sites because everyone knows they will eventually get the new intervention. It also features a built-in ability that allows each cluster to serve as its own control, providing a statistical advantage. However, Stepped-Wedge trials are more complex to design, manage, and analyze because timing and secular trends must be handled carefully. Let's bring the concept of when a great research question for a cluster trial design went wrong. We'll use a real-world example from the high-low trial in patients undergoing dialysis. This trial addressed the clinical question: what is the optimal blood phosphate target for individuals on hemodialysis? Should we aim for a higher or lower phosphate level for better outcomes? Being a pragmatic trial, high-low was set in routine dialysis clinic practice. The trial team initially chose a cluster randomization design, with approximately 80 to 120 dialysis facilities serving as the unit of randomization. Each clinic was assigned to either the high arm, higher phosphate target, meaning patients' diets and medications were managed to achieve a higher serum phosphate levels, or the low arm aiming to achieve a lower phosphate target. This cluster design made a lot of sense operationally because all patients in a given facility would be treated to the same target level, making it simpler for the staff dietitians and providers. Among other reasons, it also helped avoid confusion or contamination between patients at the same dialysis center. However, the trial did not go as planned. Here is Dr. Miles Wolfe, the trial's principal investigator, explain.
Okay, so we were 10% into the trial. This is at the 400-something patient mark. We ended up recruiting over 500 into the cluster randomized phase. We noticed this big difference in mean phosphate. That is, the high arm was greater than 6.6, and the low arm was 5.8. Now, this would be great if this was 12 months into the trial, but this was at time 0 at enrollment. We had a major imbalance. We also had a big imbalance in willingness to participate. So the low arm, which basically had to just carry on with what they're doing with no change, 2/3 of patients were willing to participate. But the high arm, which was a deviation against what people have kind of been overeducated about, that is, get your phosphate down, we had a much lower consent rate. And these were worrisome enough, and we think they were a consequence of this issue of post-randomization consent. So to be more clear, what we think was happening, patients or the dietitians thought that they were helping by saying, oh, I continuously have to bother you about your high phosphate. Guess what? We got randomized to the high arm of this trial, and you get to participate in the trial, and then you don't get bugged by me anymore about this. And so we, and vice versa, you could paint a similar picture for the low arm. So we think that there was biased enrollment based on how patients were doing and what arm they would have been assigned to, which they knew at the time of consent because their whole facility had already been randomized.
So, as a consequence of the issue related to post-randomization consent, the high-low team had to make a bold decision and pivot the trial design mid-course. Rather than continue with a compromised design, the team switched to individual randomization for the remainder of the trial. Essentially, instead of entire clinics being locked into high or low with voluntary participation, they began enrolling patients one by one and randomizing each patient to high versus low targets. This was a remarkable adaptation in a pragmatic trial It's not common to change the design on the fly, but it was the right move here to rescue the study's validity. The big lesson from high-low is a caution. If you do a cluster trial where patients give consent after randomization, you risk skewing who ends up in the trial arms. In other words, folks might self-select out of an arm they don't like, and you get biased groups. When possible, post-randomization consent or opt-in should be avoided. And if the trial intent and ethical requirements force that scenario, the trialists should be prepared with mitigation strategies to ensure the trial stays credible. Let's recap the foundation we've laid out. First, always anchor your trial design decisions in the intent and question you're pursuing. Are we trying to show something is better, just as good, or the same? Second, pick the design structure that fits your needs. A parallel design often keeps things simple. A factorial design can efficiently tackle more than one question at once. A crossover and stratification can be ideal under certain scenarios. Third, carefully consider cluster versus individual randomization. Cluster randomization can be powerful for pragmatic trials in healthcare systems, but it comes at the cost of more complex planning, management, and analysis. Fourth, identify your unit of randomization, unit of intervention, and the unit of analysis because they are not always the same. When these units are different, be sure to account for this during your trial design and analysis accordingly. And finally, be pragmatic about pragmatism. Real-world trials face real-world hiccups. Consent and recruitment may not go as expected. Logistics might force changes. A well-designed trial isn't static, it's resilient.